So, welcome to our channel. This is the episode one of the Scientist Podcast. And as we like to maintain the time in any of our interviews and seminars, let's start this session. And today we have Dr. Kanakanagavalli Shravani Prakhya, uh, who is I don't know what's your title in in your company, but she is a PhD from University of Kentucky, where I am right now, and she did her postdoc, and now she is working in a company and helping clients to make different plasmids or different nucleic acids based on the requirement for molecular biology experiments. So, Dr. Saab, tell me about yourself.
Hi, thank you so much for having me. A little bit about myself. I am from Hyderabad, India. I did my undergrad from the University of Hyderabad. I got my integrated master's, which is both BSc, MSc combined in a subject called systems biology. When I got it, it was pretty new and up and coming. We were literally the third batch, in a way, saying that we were guinea pigs. People were trying whatever they wanted on us, different
Good for science,
courses.
right?
Yes, different courses, different styles of teaching, different curriculums on us before they figured out what they wanted to do in future. So it's interesting because that was the same place I went as a kid when I was in school. We were taken around ninth and 10th class. We were taken to different universities close by for Science Day. Science Day was to celebrate Dr. C.V. Raman's discovery of the Raman effect. It's on February 28th. And we went as part of a small group to University of Kentucky, University of Hyderabad, where they showed us different experiments. And I was really, really inspired by it. And when I went back after my 12th class, I took biology because of my interest in biology, and everybody expected me to be a doctor. But then I cleared this entrance exam that I casually wrote to do my integrated MSc. This was a nationwide entrance exam followed by an interview. And when I went in for the interview just to get the idea of how an interview feels like, this was when I was 18 years old. I going in, I had people from math, physics, biology, and chemistry sitting there, professors, and asking me a bunch of questions. When I came out, I think I just came out feeling really nice. And I was like, if I get a seat, I'm going to join here. Luckily enough, I got a seat, spent 5 years in this beautiful campus, learned everything. My summers were also spent doing research because I enjoyed doing research. So I was looking at like protein and RNA import to mitochondria and looking at some malarial proteins. That's how my summer was spent. After 5 years, I knew I just wanted to do research. I still wanted to go ahead and do research. Again, that's the time where you write all kinds of exams, entrance exams like CSIR, TIFR, everything, IASC, and then I gave interviews everywhere. So I traveled like north, south. I didn't go much east, but definitely on the west side. So I finally got into IASC in my grad program for Department of Molecular Reproduction Development and Genetics. It's called MRDG. I was one of the first grad students recruited there.
So, for those who don't know what is IISC.
It's Indian Institute of Science. And if you're in Bangalore, you're supposed to call it Tata Institute. If you tell Indian Institute of Science to a Nautawala, they'll drop you at IIM, which is in the exact opposite corner of where you're supposed to go. So when we received a mail saying you're selected, they said when you land at the railway station or the airport, just don't say Indian Institute of Science. Please say Tata Institute to your auto drivers.
By the way, that has happened with me.
Oh, you did?
Like a long time ago, I went there for a KVPY interview,
Oh,
and we were just like circling around Bangalore. But yeah, so I can understand what you mean. Yeah, yeah.
yes.
Okay.
Yes. Um, Yeah, I spent a couple of years there, and due to some family reasons, I had to quit my grad program there and then start over and reapply for grad school. Then we were looking at some umbrella programs where you could do rotation and figure out who you want to join with during your PhD. And University of Kentucky had one of those umbrella programs. It's called Integrated Biomedical Sciences. I got in and I did 4 of my rotations. Two of them were in neuroscience. One was in hearing, and the fourth one is where I joined, which is in a platelet lab. We were looking at blood clots, hemostasis, and thrombosis. That's where I got my PhD from. During my PhD, I think every stage I was like, I want to do more research, but I think during my PhD, I realized that I enjoyed talking about science more than the doing of science. So, and also I wanted to move to an industry.
Hmm.
I sat down and applied for as many jobs as I could. And currently, I'm a technical account manager at a contract research organization called GenScript. As Abhilash mentioned, I help my academic and industry clients with anything from molecular biology, plasmids, mRNA, peptides, proteins, antibodies, anything they want. I help design the projects.
Hmm.
And I'm like the primary point of contact between my production team on the company side, sales, production, marketing team versus my clients.
Okay, that's a very nice introduction. Too much information to unpack. And those who don't know about these things, please go and Google IISc. That's the number one premier institute of India that does as good as research as anywhere in the world. Okay, not only in the India. GenScript is one of the largest country now, company now. Those who survived COVID and everything, and is still on the top in doing business. And many people think that scientists are in science is in university, but industry is for business. So if someone is scientist in industry, what does that mean? Is there any difference? Okay, science is science, wherever you do.
Yes, there. I don't do research in industry, so I cannot speak for scientists in industry. But little information I know, I think they're doing the same kind of science both in academia and industry. It's only that the science in industry is more driven by a certain kind of a result or a certain drug target they are testing. So it's more channelized and they know what the next step would be. And if it doesn't meet the next step, they would find like a different area to work in. But I think it's more wider and you're not very results driven in a way that you don't want this result, then we are scrapping the project kind of situation in academia. You're probably still chasing and saying, why it is not this result.
Okay. Mrityunjay, do you wanna-
Yeah, so first of all, thank you for joining us today. And I think so. What's my interest within industry is that how do you guys actually figure out, right, as to okay, this is the problem that we want to solve. I mean, or like because you've just mentioned that you guys are more interested in, hey, this is a problem, we can solve this, and obviously we can make some money out of it, and that's what industries do, right?
Mhm.
So what is the process of actually figuring out what problem you guys want to solve?
So for me in my current job, it's usually very client driven.
Hmm.
We have a certain things that we definitely do, but something else that we can't do and the customer asks for it.
Mm-hmm.
We go to our marketing and production team and let them know that this is what the client wants. Can we do this on a small scale, or can we do
Mm-hmm.
like a beta testing with these guys? And
Mm-hmm.
once we know we are capable of doing it, we can scale it up.
Mm-hmm.
So usually it's driven by, at least my company, since it's a contract research organization, it's more driven by what the client wants. If they want a certain things, we try as much as possible to accommodate their needs,
Mm-hmm.
even if it means we have to go around and tweak
So
a little bit.
very intriguing. So, and I'm assuming because you work with academia, right?
Mm-hmm.
My question would be like, what is it that you guys have, right? And you guys can do that academia can't do. So and that's why they come to you guys.
I think everything we do, academia can do. The only thing we always promise is, you leave the more mundane stuff to us.
Mm-hmm.
Like cloning, come on, give it to us. We'll take care of it while you focus on the bigger problems that you want to solve. So we don't want we don't want a customer to be stuck for 3 months on making an antibody. I can make it for them. My company will make it for them. Or 3 weeks on a clone where we can deliver it in 5 days.
Hmm.
So
So,
that's what they come to us for, something that's very quick, and they have to not think about it once they place an order, and then they can move on with the bigger problems to solve.
I think this is one of the nicest answer.
Yeah, I think what I can see is that industry, can provide speed and efficiency,
Yeah,
right?
it's
Mm-hmm.
like a best
Uh,
aid for research.
Right, and I think that's also really important,
Mm-hmm.
especially when we are trying to do something that has some sort of a time limit, right?
Yes.
And a good example is, what happened when, COVID was there.
Mm-hmm.
Everything was on a time limit, and I
Yep.
think industry can always, be a little quicker than academia, right? Can be more focused than academia.
Yes.
Shravani, I mean, it's very interesting. I know that you work as as a technical account manager, but I wanted to also maybe quickly understand, right? Like in your day to day, right? What kind of problems? And you don't have to. I mean, obviously there is some sort of like client and business kind of space where you can't reveal certain things, right?
Mm-hmm.
It's okay, not a problem. But is there anything, maybe you can take us through your day to day, right? How does a day look like in your life in industry?
Yes, my day is going, every day is going to look very different depending on what problems my client come with or the new
Mhm.
projects they come with. Suppose I have a very, very complicated project that comes in at 10 a.m. I'm probably stuck till 4 p.m. just doing that.
Mhm.
So it depends, it just keeps changing. But usually what happens is my sales or the customers directly reach out to us and
Mhm.
say, we need you to help design this project, and they
Mhm.
give us the project details.
Is it
And
possible if you can maybe take us through one project which you guys have already done like maybe a couple of years ago and you can talk about it?
oh, yeah, sure, it can be some sort of a cloning project, or they say,
Mhm.
oh, we need
So
this gene
when
cloned
you
into
say
this.
when you say cloning, what do you
Yeah.
mean, right? As a layman, I feel I'm cloning.
Do you make new human beings? Then
No,
you are
I
the
Right.
wish
uh,
I wish we did. But it's usually they have a gene of interest that they want
Yeah.
it to be cloned into a vector of their choice.
Yeah.
So
So again, a lot of things that is quite obvious to you, but maybe if someone who's maybe listening to you, maybe they don't know, right, what you're talking about. So
female.
maybe a little detail as well, if that's okay.
yes, so if I want to express, let's say, a green fluorescent protein
Ooh.
in my lab, and then it is going to glow green when
Mhm.
it's been expressed. So they give me the sequence of the green fluorescent protein, and they put it in a plasmid that enables them to actually express it and glow.
Mhm.
That then I take the sequence and I put it in a way that I make sure that it's the right protein that is being expressed. And once they take it to their system and they stick it on their plate or grow it in the culture,
Mhm.
it's going to glow green for them because we have expressed a green fluorescent protein
Yeah.
successfully.
Yeah.
More often, they usually more complicated projects that's going to take really a lot of time. These are mostly, it's our bread and butter, and we don't take as much time because we just it's just playing and solving a puzzle at this point. The more complicated ones would be like designing an antibody for a future therapeutic use, or they are trying to use an antibody as therapeutic and test it in animal models. So just designing those, making sure all components of the antibody are correct and rightly going to be expressed, that's something that's going to take a little more. It's again also puzzle and problem solving, but it has more steps
Hmm.
along the way.
Hmm. How's your life in industry? Is it boring? Is it exciting? Like, what do you guys do for your lunch breaks or your tea breaks?
So my job is a remote job, which is something
Ooh.
that's the only part that I am not the most excited about because I love meeting people.
Yeah.
My lunch break happens on my desk, so I just eat lunch at my desk, thinking about some
When they
emails
call
to
you,
respond.
I was like that when they call you on site.
When they call us to our office, our lunch break is mostly socializing. We just
Mm.
sit and take a nice lunch break and uh, socialize and talk about everything other than work. Or if we have a very interesting client, we just say, hey, this is going on.
Hmm.
Any ideas are welcome. Um, otherwise it's just what's going on with your life, just catching up, because we go to office like once a quarter, so it's just catching up.
What do you mean by interesting client?
Oh, sometimes we just have things that are going on, like there's
Hmm.
a lot of back and forth or something like that. Then we just discuss it in a way to come up with, the most optimized solution. If I'm in a problem,
Yeah.
what's the most optimized solution to help them?
Hmm.
Uh, And so you have a unique situation that you have done research in your undergrad, in your master's, both in
Hmm.
India in two different places. And then you have done research here in
Hmm.
University of Kentucky as a PhD and as a postdoc. And now
Yep.
you're doing research related things in a company. So you have
Yep.
multiple city, multiple country, Saar Samandar Par experiences with research.
Yes.
So tell me some positive things about research in India or in US.
Sure. So I think the best thing about research in India is the community. It's so tight. Like even different departments, your friends are very tight. You're sitting in the mess and talking about again problems. Like, my gel didn't run properly or something didn't work out. And then you're coming up with solutions at lunch. So which is something I miss. I just miss those scientific conversations, because here, if you're meeting your same lab for lunch, you mostly are trying to discuss about what movies you've seen or what shows you've seen, because it's outside work. But in India, I think you take that being a researcher very seriously, and you build a really nice community of friends there. They'll have your back. They'll do anything for you. And when they defend, also, it's like the whole community comes together to cheer for you. It's just awesome. I think that's the best part.
One
Just
example.
one example. I did not defend one example we have in front of us.
Yeah.
I think he carries Abhilash carries that camaraderie, that community feel even here. I still discuss. I think when Abhilash showed up here, I think I found a really nice friend to discuss science with outside my lab. Also, it helps me a lot to get some things off, or at least know a third person's perspective. Like, he's not from my lab, but he can give me a perspective that can help me.
Why do you think talking to someone else about your science is really important? Do you think it helps, or I mean, is it something that comes from an environment of, let's say, high pressure, or is it something that you just want to do?
I i mean, for me, science for me, as I define it, is to unlock the secrets of the universe. If it's so big, you want to talk as much as possible.
Yeah.
And just knowing, as I said, the more you talk, the more people will come up with different ideas, and ideas don't come in isolation.
Mm.
So just talking about it really helps. I mean, there can be something that Abhilash would have told me about my research. It would be like, oh wow, I didn't think about this, because I was too busy and narrowed down in my
Mm.
thoughts,
Mm.
that somebody else just throwing a random idea can
Mm.
actually change the course of your project in a really different way.
Do you think it can happen with researchers to have like a tunnel view when they are, when they're working in their PhDs, postdocs? I know Abhilash had a different question. I just want to ask this last
Oh,
one. Yeah.
that's all right.
Sure. I definitely think when you're chasing a problem, I think you get too lost in the minute details of the problem. And there are times you just have to sit back a little bit and look at the big picture or the bird's eye view. And when you're chasing a problem and looking for a solution, you definitely become tunnel viewed, because you're just like, this is not happening, that's not happening. That's why you need someone to sit at back and be like, have you tried this? And that's when real solutions, you because you're so lost, and you look at so many minute details of it, you just lose the big picture sometimes. Which is also why I think people, the whole idea of presenting in conferences and somebody else asking a question from a different part of the world, it's just like, hey, have you thought about this? And it helps you take a different course for your project.
Yeah, yeah, that's really nice. And anything you liked in US that really pulled you like, okay, 5 years, I'm
So
so
India, its community, what's US, I
oh yeah,
guess.
what's US?
US, there are too many things. I think it the the relationship between your PhD mentor and you is very different in India compared to the US. In US, they
You're
treat
saying
you
it's
like
not toxic?
no, no, no. It is very it's like a peer. They look at you like a peer. They ask you to call them by their name, and your ideas are more valued here. You just can throw a random idea, and they will not be dismissive about it. They just like, oh, you figure it out and come and tell me about it. So I think that whole idea that they let you be and you can throw anything at them and they're very receptive to it, at least that's how my PhD advisor was. So that was the difference I found. A very strong difference. And I think here we have a better work life balance because a lot of people take even research as like a job. It's
Hmm.
not a 9 to 5 job, but people do take it as that, or at least you get your weekends off. In India, as I mentioned, it's such an integral part of your life. Even your look, like I would put a PCR reaction and go watch a movie and come back and run a gel. So it's just even your off work depends on your work. So
Do you
it's
think do you think sometimes it's also because of the glorification of working after hours in India?
It is. It is. Because my PhD advisor went to IISc, Indian Institute of Science, when he visited India a couple of years back. That's what he told me. He's like, it's 11 p.m. and I see the lights on in labs. And I was like, yeah, that's what I was doing. I had a sleeping bag in my lab and I would run experiments and stay back at night. So it's over glorification, which is not good for your physical and mental health.
For
Absolutely.
your circadian rhythms.
For
Thank you.
your circadian rhythms. I
Thank you.
think Dr. Prabhat can give you
Thank you.
more
Thank you.
details
Thank
on
you.
it.
Thank
Always plugging his
you.
own research.
Yes.
And okay, so we have talked at least I think it has been 15, 17 minutes, so that was great.
Yeah.
Quick question: do you have a favorite scientist, and do you have a favorite discovery of any science?
Favorite scientist, yes. My favorite scientist is Rosalind Franklin.
Yay!
I loved her work. I felt like she deserved more than she got. She's really my favorite scientist and somebody I am very, very inspired by. The scientific discoveries, I think we are in the best time for scientific discoveries. There are lots of cool things happening, but some of them that just comes to my mind right now, which I think are going to change the face of therapeutics, which has already been changing the face of therapeutics, is one is CAR T therapy. I'm so inspired by it. And the other one is CRISPR. I'm very curious to see how CRISPR is going to change, is in the process of changing
For those who don't know,
therapeutics.
CRISPR is gene editing tool where you can mutate any gene, and then you don't need to do thousands of different experiments to do it. And Dr. Dodna already got Nobel Prize for it a couple of years back, and you should read her things. She's absolutely incredible. It's a gene editing tool.
Mm-hmm.
You don't know should know that. And do you have any final question? So we
Yeah,
can.
yeah, I do. I do.
Yeah.
Sorry. I am going to ask this question because you're talking about Rosalind Franklin, right? And you said couple of things, right? You said that she obviously you like her, right? So what I would love is I would love for you to tell everyone what does she do? Like what was it that she invented? And I think you also mentioned that she didn't deserve, right, what she was supposed to deserve. Maybe a little light on that. And a segue question to all of this is, do you think this is something, and again, like I'm not asking this question from a point of view of hatred or anything bad. I'm asking this from the point of view of an observation, right?
Mm-hmm.
Do you think there are a lot of Rosalind Franklins, right, who are still there, who still exist. And is there something that is happening right now to maybe stop that? And is there something that should be done? So that would be my final question.
Sure. So Rosalind Franklin did the experimental work for the DNA double helix structure.
Hmm.
The Watson and Crick, who got the Nobel Prize for deciphering the structure of DNA, their work was entirely based on the X-ray image that Rosalind Franklin had generated. So ideally, I think she is as important part of the Nobel Prize as they got. So she should have been the third contributing member for the structure of DNA Nobel Prize, but unfortunately, she did not receive the Nobel Prize. But also, because of exposure, she died a very early death due to cancer. So I think the fact that she's amazing at what she does, which is what inspired me to be a scientist. But your second question is, there are many Rosalind Franklins out there, which is which is sometimes really heartbreaking that some you don't get enough recognition just because your of who you are. You're a weaker sex, or that's what you're perceived as, and you don't get as much recognition. But we are still breaking barriers every single day, and I don't think being a woman or a man is going to decide how good you are at your science. Another example of somebody who had to go through a lot and still make it is I can't remember her name, but she got the Nobel Prize this year for the mRNA vaccines.
mRNA, yeah, yeah, yeah,
Yes.
yeah,
Yeah.
So I think she made it a point. I really admire the fact that she made it a point to write a book and let everybody know her journey.
yeah.
So we are well aware of what happens and how administrative things work, and how you can actually, how you can actually work to break these shackles and move
Those
ahead.
those who are in biology, they know another example called as Barbara McClintock.
Hmm.
So she discovered jumping genes. So there are some genes
Transposons.
who jump and go and bind to another place, and that's very important for all biology, like whatever we are.
Hmm.
He discovered that in 1940s and 50s. No one believed her. She left research,
Hmm.
and then scientists discovered, oh, she was right. And then she got Nobel Prize in 1983. So after 50 years after her discovery, no one believed her. So so these things happen a
My
lot.
final question, Shravani, would
Yeah.
be,
Yes.
so let's say if there is a young, researcher, men, women, doesn't matter, right?
Mm-hmm.
Because that's what we want. We want an equitable, research and science for everyone, right?
Mm-hmm.
If there is a young researcher who wants to do PhD in the US, right, or who wants to get into an industry wherever in the world, like, is there anything that they should do? Like, what would be your suggestion to them?
For getting into a PhD, I think just being passionate about what you want to do, know your whys, because not just getting into the best part of getting into a PhD is getting into a PhD, and then you know what happens. And there are your lows when you hit rock bottom. Just knowing your why, am I doing this, and being very clear about it is the one that's going to get you out of your rock bottom.
Hmm.
So know your whys. I think your why should be as good as, I want to cure cancer. I want to cure diabetes. I want to understand how the brain works. Your whys are very important to you. It's very specific to you, and they can be a personal reason why you want to do research. But just knowing why you want to do research is so important, because that's the one that's going to get you out when you have a bad day of experiments, when you have a bad period where your experiments are not working. Going back and remembering why you started this in the first place is going to get you out, and that mindset is only the one that's going to drive you to the end of your journey.
Wonderful.
Moving to industry, I would say it's just network. You are supposed to network like there's no tomorrow. And most people start networking when they need a job. And the best time to network is 2 years before you need a job, which is, you don't know. But the best time to network is every day.
Hmm.
So people have to update their LinkedIn and not be shy to put in their achievements. If you've done something, you got a grant, you have to go and put in your achievements. It's not because you are a show off or anything. It's just, one, you're tracking everything, and it might be very easy to write your resume later, but
Hmm.
also you worked really hard to write a grant. It just didn't appear out of nowhere. So you have to put in your achievements. You got an award, please go in and put in your award. So have an updated LinkedIn, talk to people you like. Most people, alumni are the best people to network.
Mhm.
and just saying, oh, I was a fellow IASEN, they will want to talk to you. I am a fellow wildcat, they will want to talk to you. So always have a really nice network, and they're the ones who want you to succeed. They want you to go ahead in life. So they will definitely try to help you.
Thank you so much. It was great having you. Obviously, this is our first, so we started something, and I hope you are not bored of our questions and our.
I had so much fun. Thank you so much for the
And
opportunity.
and someday, if anyone in Hyderabad, your hometown, is gonna watch this, and anyone is studying in Telugu, so do you wanna say in Telugu something to your friends who are in Hyderabad who want to pursue science in future in a minute, if you want?
Oh, I haven't thought about that. Uh,
Your
I don't
language.
know. I don't know. Yeah, one line is Baga Chadavanti, which means
Okay.
study well.
Oh, that's a great line.
Yeah.
Okay. Thank you so much. I'm gonna turn off the recording just for a minute.